Journal clubs are actually my favorite event on the sort of regular DiMe program. It's such a nice opportunity to bring together a smaller group of experts in our field who are particularly interested in a topic and pair them with a terrific scientist with some recent publications to share with us. So let me quickly, before I go ahead and introduce our discussion lead for today, Eric Daza, let me go ahead and just run through some housekeeping for folks who may not have joined a journal club before. So as you heard, as a voice from Zoom announced, we are recording today's session. The slides and recording will be available on the DiMe website, if not later today, then certainly tomorrow. So Eric, your expertise and the content of this conversation will be available to everyone. Journal Club is a more informal meeting. It is primarily discussion based. So Eric's going to give us a little bit of some of his insights from behind these two terrific sort of posts that we shared. And then it's really over to you all to sort of guide the conversation, to ask good questions. And you can do that in a couple of ways. You can either raise your hand, not like this, although I guess I would see you, using the raise hand function, or if you prefer, you can pop a question into the chat, and then we'll call on you to ask that live. So that's a little bit of housekeeping. Look forward to a great discussion. But before we get there, let me go ahead and introduce, or ask you, Eric, to introduce yourself as we think about statistical considerations for successful digital health innovation. Eric, tell us a little bit about yourself and your background, and then let's go ahead, dive in and hear some remarks from your side before we enter discussion.
Great, thanks, Jennifer. Hi, all. Great to be here. I really thank the DiMe Society for having me. So my background is I was a biostatistician in pharma for 5 years, a master's level biostatistician. Before that, I was actually a neuroscience, neurobiology major, so I wasn't even statistical as an undergrad. I worked in pharma for 5 years and then decided I wanted some more of the statistics pie. So I went back to school for a doctorate in biostatistics, a DRPH, Doctor of Public Health. That took me about 8 years. I got a lot of experience with public health applications, survey sampling, global health, maternal child health and nutrition. And then apparently I wasn't done with academia yet. I ended up doing a postdoc for 3 years at the Prevention Research Center at Stanford, which is a behavior change center. So that's really sort of where I got introduced to behavior change type thinking and concepts. And while there, I started getting interested in wearable devices and what I could do with that. So on a conceptual note, I got interested in N of 1 designs, or single case or within patient designs, where you're trying to figure out what the patterns are for one person over time. And then, so all of that, I'm getting a little better at this elevator speech and compressing it, but that leads me to Evidation. I was at another startup prior to Evidation, where I learned about data science and healthcare claims data and how to deal with that sort of, those sorts of challenges. And at Evidation, it's awesome. I mean, I get to work on device data on behavior change, both from wearable devices and from patient-reported outcomes or surveys, which are administered through our app, and help organizations and companies kind of design and run their studies from there as a statistician. So it's awesome. I should say as a data scientist now. I'm both, but definitely in the data science team. So that's me.
Fantastic. Thanks, Eric. Do you want to go ahead and share your screen? Take us through a little bit of background from your side about some of these terms that we're trying to debunk, and then we'll throw it open to questions. Sure.
Can you all see my screen now?
Yep, we got you.
Awesome. Okay. So that's me. I'm going to take you through basically the main points of these 2 blog posts I wrote. But before I do that, I'll give you the academic imposter syndrome disclaimer and say I am not at all a deep expert in either of these, in like the articles I cite and what's been done to rectify these issues. I'm speaking more as somebody who's out there in the field who's been trained deeply in these concepts. And so I like to bring up where I see these concepts kind of flagging or in need of improvement out in the field, both in traditional research and in digital health. That's my little disclaimer. Protect myself. So the first of 2 articles I wrote was called Artificial Intelligence. If you've read any of my ramblings online, you'll know that I like to play with words a lot and takes up an inordinate amount of my time when I write these things. But the idea here was that I was thinking of this concept called harking, which I'll show you in a second. And the idea is that you should report your modeling strategy or statistical analysis plan before seeing any data. And it talks about the importance of that, because if you don't, then it's harder for you to keep track of how you might be seeing patterns that don't exist in your data. You just keep looking at it, you keep modeling over and over again. Before you know it, you're finding something that only exists in your data, and then you try to use that model on another data set and it doesn't work. So some of the key points involve, well, here's the definition. Harking is hypothesizing after the results are known, while claiming that those hypotheses were created before the results were known. And usually this isn't intentional. Like we all do this all the time. I do it too, and I'm a statistician, because it's so hard to keep track of when you started looking at your data and then adjusting your model. Every time you hark, you risk claiming the implausible, that you reproduced a signal when in fact there was only noise. And the idea here is that if you engage in harking, you're confusing a few things. You're confusing ideation or discovery with iteration or replication, discovery for confirmation, and you're seeing your in-sample performance, but you're accidentally thinking it's out-of-sample performance because you thought you used your holdout set, but then you went back and adjusted your model to get a better fit on your holdout set. Voila, your holdout set is now part of the training set. And so I kind of made up this term, inside out of sample performance. I do really love the movie Inside Out, so that's part of it. But the idea here is that you may mistakenly flip your interpretation of your model's performance from in sample to out of sample, so inside out. If you'll tolerate my wordplay a little more, this one is actually less wordplay and more kind of directly, here's my recommendation. So I wrote this sort of catchy clickbait title, Dish Statistical Significance. I hope people click on it because they'll see the subtitle, but keep statistical evidence. Because I am not advocating for getting rid of statistical evidence. It's literally just the phrasing that I have an issue with, and a lot of other statisticians do too. So the idea here is that statistical evidence isn't the problem. The problem is what the simple alchemy of transmuting statistical significance into implicitly scientific significance creates by dropping just the one word statistical. So it feels like we have scientific certainty in some way, even though it's not there. And then the last two bullet points really are just, I thought of this catchphrase based off of, you've heard the catchphrase, absence of evidence is not evidence of absence. Well, this is very similar. Significance of evidence, so in this case, statistical significance of the evidence, is not itself evidence that the thing that you're trying to estimate is scientifically significant. So that's what this phrase is meant. And if you think about it in terms of the original phrase about absence of evidence, it actually comports really well. They kind of make sense in very similar syntactical ways. And a little explanation here, what I'm talking about is that a significant p-value or statistically significant p-value is what I would say corresponds to a discernible finding, which means you have enough, your sample's big enough and the difference is such that you can start to really kind of discern what that true mean is that you're after, what the true finding is. Or you can't because your sample isn't big enough or the mean is too close to zero. So that's why a finding that's discernible corresponds to a statistically significant p-value, which is why I like using the word discernible instead. And then the last plot I'll show, I just created this yesterday. Don't get too caught up here with the specifics. This is just a schematic. It is in no way intended to convey any sort of actual metric. But the idea is that if you have, from your data analysis and exploration, you have confidence intervals and p-values, which are measures of statistical significance, or what I would call discernibility. Well, here, if you think of the y-axis as like the value or usefulness of what you can get, and you look at the x-axis, that's the number of looks you're taking, looking at your data. So when you look at your data the first time, the value that these metrics, CIs and p-values, give you for generalizability out of your sample is pretty high. And if you're using it to say to discover insights, it's pretty low because you really just have hypotheses or models you had from the beginning. You're not really discovering anything. The more times you look at your data, you start to say, oh, there might be something here. So the potential for discovery starts to go up. But as that model gets tweaked, the ability for you to use those metrics, CIs and p-vals, to generalize starts to drop, because now you're starting to say, basically the calculations for CIs and p-vals, they don't know how many times you've looked at your data. So they're just assuming like, okay, well, the analyst, the data scientist is giving me data. Here's what happens if we compare this to another data set that looks like this. It has no idea if you started looking at it once or twice or 10 times. So it starts to lose its ability to be a metric for how well this finding generalizes or how much you can infer into reality. So that's it. I already just dumped a whole lot of concepts, so I'm going to stop there, and maybe I'll stop sharing now, or I could keep this up. Jennifer, it's your call.
Why don't we stop sharing, and I'll invite anyone who's somewhere where they feel comfortable putting their camera on to do so and sort of join the conversation. I will look for, oh, fantastic. We already have a raised hand. Let me see here. Ansar, go ahead.
Hey, Jen. Hey, Eric, thank you so much. I'll admit I read your articles this morning. And as I was reading through them, I was like, yeah, this makes total sense. I completely agree with all of this. This terminology is often misinterpreted. And then I'm at the end of the articles, I'm like, okay, well, I agreed with all that. So I was probably not the target audience of this article. Who were you writing this for?
That's actually a very good and deep question, because recently in general, I've asked myself, what's the purpose of me writing a particular piece? Is it mostly just to rant, or who am I trying to target? And these ones, in retrospect, I think they were written for, it's more for folks, I guess, less like you, who are not quite aware that these are issues that are important to handle, specifically the statistical significance ones. I think, so I'll split this up. I didn't quite have a clear audience in mind for the artificial intelligence piece, just to kind of put that out there for folks who weren't aware of that being an issue, because I don't really present any sort of solid solutions to that. It's more of an open question, like, this is my thoughts on it. If you're interested, give me a thread back on, well, that's great, Eric, but we can't do it for what we do at work, or, oh, that's great, this is how we do it, and then maybe we can use that. For the second piece, that was really more targeted towards, I don't want to call out any particular professions, but there are some professions that hew to using significant a lot instead of statistically significant. And it's really kind of targeted towards that audience. And it's more of a, hopefully I put in the article, I know this is, I'm making this recommendation, but please give me pushback. Like, why won't it work? For example, reviewers will give me pushback if I write discernible. So how can I sort of help convince reviewers in my papers? And that's the purpose of that article. It was to give readers example text that they could then use to change around for their own publications. That's straight lifted from my own work. That's text I've massaged a little bit from my own work that I use to give to reviewers. And then if the reviewers push back, there's only so much you can do. So it's like, but I'd like to give people a fighting chance to change the terminology if they agree with sort of my take on that. Does that answer your question?
It does. Yeah, thank you. I mean, I thought that this is definitely like the people that I would think this kind of information would be really helpful for. It's definitely like, I just think we should all kind of stay aware of this, because it's easy to fall into habits. I think it's probably pretty important for individuals that take results from scientific articles and try to communicate them to a broader audience to be more aware of like making that jump from calling something that was statistically significant to something that was significant, which I think is a really important mistake that a lot of people make pretty often. So thank you for that. I thought they were both well written and enjoyable to read.
Thank you, I appreciate that. One last point is this is also, it's also speaking to statisticians, because we're trained to shorten statistically significant to significant, so it's not like a statisticians telling non-statisticians thing. It's more just like, I feel like this is a change in practice that as statisticians, we're not trained in things like behavior change and habits and cognitive bias, but because of my undergrad and a little bit of my work in my postdoc, I started to realize these sorts of things like p-values and significance, they aren't really technical issues. They're behavior change issues. They're habit change issues. And that's, I think, why statistical pedagogy has struggled to recognize and help folks understand that who are practitioners.
Andrew, I love that question because, I have a habit, Eric, of writing for me instead of writing for the reader. And with this in particular, who are the change agents here? I thought that was terrific. Volodymyr, you've got your hand up. Do you want to ask the next question?
Yeah, thank you. Yeah, great articles, Eric J. So actually it's related a little bit to Anzar about your first one that you presented today. That article actually was written just for me. I read it, and I'm a data science professional working with data and working with like test and learn every day. And funny thing happened like about this harping. This is correct? I'm pronounced harping.
Arcing, yeah.
Yeah. Of course, the next day somebody came to me and said, like, "Oh, look, we run this like experiment. We're looking about for difference in envelopes and how people are reacting in those envelopes. So we found no difference, but surprisingly, there was difference in calls, and this is really important afterwards. Like when we look at the data, and I was like, I almost fell from my chair. Like I just read about this day before in your article, so." The question to you, but I was thinking about this like all over again and again, like, so what are the pitfalls of that approach? So let's say you design an experiment to find one thing. After you analyze all of the data, you found something else, but it looks like it's still valid. I was trying to think about this because you did your randomization, group A, group B. Group A has a bigger increase in calls than group B, and it's like significant, or let's say it's statistically evident, it's important, it's a big difference. So what are the pitfalls of like saying that you found real difference in something that you did not expect to? Like this experiment was not tuned for the calls, like increased number of calls, it was for something else, for like envelopes and reading the letters. So what are the pitfalls?
Yeah, that's a great question. Volodymyr knows this is coming, but I have to disclaim that Volodymyr is one of my dear friends and colleagues. We used to work together at the startup before Evidation. And I was expecting him to give me pushback on the significance article, but
no, but that one was like right on point. Like I was very surprised. Like it's right next day. I'm telling you, right next day I read your article. It came to me like this problem just presented itself.
Yeah, pitfalls are it's hard. The pitfalls are that it's almost like once you start moving away from the original hypothesis or test or question, we don't really have really good metrics to start to determine, oh, how well is my new finding going to generalize, which is totally okay. That's fine. It's just that I think we, as a society, we get so enamored with, or maybe hateful of CIs and p-values that we start to use them for everything. And they're really only good as measures of how well does this generalize for the initial question. All the questions past that, like, oh, well, maybe if I look at this subgroup, oh, it's higher here. Now, we're not sure that if that will generalize. It might, but we need another sample. So the pitfall is like, we literally don't know anymore how well it will generalize. It could, but the literal answer, statistical answer would be, we need another sample to test anything after that first look. What I want to say though is, on top of that, I feel like the culture in statistical sort of pedagogy and training has been just horrible for exploratory science. I mean, we're trying to tell people, here's like CIs and p-values, and you use them once at the beginning, but that's not how science works. Science is like half exploration. I mean, maybe more than half exploration to generate new insights. Like, oh, maybe it works better in this subgroup. Well, the bulk of the methods we've developed are for replication, not exploration. So I think that's sorry, that was like a long answer. Volodymyr's used to this, but I apologize to y'all who aren't. But the pitfall is that it's hard to tell right now how well those things will generalize to another sample.
So
I like to.
Your recommendation would be just to run a second study, right? And then tune that study, like calculate the sample size, but then for increased number of calls, right? Like that difference that you're looking at, maybe different sample size groups, and just do a follow-up study.
Or even, and I think Volodymyr, I think you and I have talked about this, but in data science, we have an abundance of, we have like what's the word, something of riches, I can't remember the phrase, but
embarrassment.
There you go, embarrassment of riches. Thank you, Jennifer. It truly is, right? Because you have all these data and we want to use all of it to find insights and patterns. But Volodymyr and I have talked about, what if we just use like samples of that data first and parse it out? They're still going to be pretty big, but they'll random samples. Then we don't have to collect an entire new data set. We can just use the next part as the next sample to test those discoveries we've made in the smaller first sample. I think as human beings, we want everything, so we want to use it. So it's kind of like, oh, I just like struck it rich with this like load of oil or whatever back in the old days, crude oil, and you just go through all the oil. You have no concept of parsing it out and rationing it because you feel like you should use everything you have in front of you. Anyway, maybe that seeded more questions, but yeah, Volodymyr, great question.
Really terrific question, and I like the idea of applying a little bit of a sort of parsimony to the data on deck, especially as volumes and sort of velocity are certainly increasing. Danielle, you put a really thoughtful comment in the chat. I'm not sure if you're somewhere where you can unmute yourself. I'll give you a second in case you can, because I'd love to hear your thoughts on sort of the value of some of Eric's pieces.
Sure. Thanks so much, Eric. This is such a helpful discussion. I too spent a long time in academia, and there is always that pressure to find significance a lot of the time. And, I think there are a lot of trainees who feel like, well, if I don't find something significant in this data set, how will I move on in the field? How will I publish? How will I get to that next step? And so, of course, this is a resource that's helpful for scientists, statisticians, everyone who's working on data. But I think it's also super helpful for trainees to start early on thinking that it's not just about finding something that met that, statistical significance value, or just, I hit 0.049, I'm good to go, I can publish now. It's just, I think, really helpful if you start early on, and if they can hopefully see these articles early in their career to help them know that, it's not important just to put a paper out there because you found some number, but really, what is the meaning of this? How does it generalize? And all the important points you brought up already.
Thank you, Danielle. I love that. I love hearing that too, because you actually, you're speaking to a really important point, which is that it's for me, it's a culture habit change issue, right? And that's less likely to happen with those of us who are already out in the field, who have our old ways, and we're trained to use significant and all that stuff at 0.05. But the trainees and the students are the ones who are going to be moving that change forward, because you're still going to be taught that in classes, but at least if we can sort of see this idea like, oh, okay, this is the way to think about it. And as you move further into your professional careers, you'll be able to advocate for changes in practice much more so. And you'll have the intuition behind it that we didn't really have when we were being trained. Yeah, so thank you.
I really like that, and I want to go back, if I can, and a little bit to sort of Amzar's question about sort of who were we writing this for, because certainly as a reader, I'm not a statistician or a data scientist, but I was, everything, Eric, that you sort of stated resonated, it's sort of jiving with other experts on the line. But as a consumer of findings of this data, like, how do I know what happened, right? You talked about some of the solutions potentially being around reporting. Like, if I want to look at published findings, or if I want to look at claims based on these sorts of approaches to data, how can I, as a non-data scientist, figure out what the heck happened and whether the findings are trustworthy, generalizable?
Yeah, it's so the two-part answer is with respect to statistical significance, that might be I don't know if it's easier, but the text for that is already in published articles. So it's a separate but related issue to harking, right? With the language of statistical significance, you can look in an article, and I don't know what the training would be, but it's something like if you see a claim that there's a significant effect of A versus B, well-written articles should have a place where you can look back to see, oh, what did they mean by that? Oh, it's actually very small, but their sample size is 5 million. Okay, so that's a flag that yeah, there is probably a difference there, but it's inconsequential in real life. So you can look at, I'm not giving a great answer, but at least for the language of significance versus discernibility, there's already a path forward in some sense. I feel like it's much harder with harking. One of the ways that psychology and biomedical science, so clinical trials, have tried to address that is, and a lot of journals have too, is through pre-registration, where you have to report your hypotheses and your models you thought of first, what are called a priori hypotheses or models. You have to report them first so that the public can see, oh, okay, these were your original ideas. So whatever you report that you found in trying out these original ideas, those are the ones that probably hold somewhere else. So they're probably more general, more true. But then everything else, they might be true, but we need more evidence. We need another study to see. So pre-registration is kind of the short answer, I guess, to the harking piece.
And you said that one of the things I noticed in the article, you said, something like clinicaltrials.gov might be a really good place to do that. Also, you mentioned that sort of journals are increasingly requiring it. Like, is this being established slowly as a requirement, or are we not there yet?
I don't know if other folks on the call can answer and can chime in on this as well. What I know is that I think there's a slow culture shift to making this maybe a little more standard. There are journals that I can't think of them, but I read recently. There are journals that, or there have been articles written that have tested this in this sort of wonderful meta analysis way. They've tested out pre-registered articles and their quality versus non-pre-registered articles and their quality. So yeah, if anybody else on the call wants to hop in.
I think in regular academic research, I don't see like a platform. I think it definitely, for me, it's come up in the review process where a reviewer's been like, you've listed some findings here. Can you actually separate the ones that you had a priori and the ones that you kind of figured out during your like exploratory analyses of once you were looking at all your results? And it was nice for that to come up during the review process. I mean, the good news is in clinical trials, at least they have to put down a protocol and a statistical analysis plan and lock that in before they actually look at the data. So that's good, at least for drug development. It is pretty formally defined in advance. But it's also just like a cultural shift, as you're saying, because I mean, we're talking about, don't wait for the 0.049 if you're a grad student, but like the grad student wants to graduate, and like they want to publish, and it's not their fault that the journal's not going to take their paper because of these like non-significant findings. So it's actually a much bigger problem. And I feel like maybe this is something that editors and reviewers need to be more aware of. We should publish negative findings if we can. And that's a whole different conversation. But it's very related to this topic is the data is the data, and everything is sort of can be communicated to some extent.
Yeah, and I think that was, you're reminding me again, Anzar, about like the purpose of why I wrote that piece was to really put it out there so that folks had text to use already. It's, versus like, say, like somebody like you who's like, oh, I totally agree with these concepts. Great. Okay, now I got to finish this paper. And how do I,
yeah.
Oh, I got all this other stuff. I got kids to take care of, whatever. Like now I've given you template text that you can now copy, paste, and then modify. And that was the hope.
I thought that was really well written, by the way. To be honest, where I think this is really helpful is particularly in healthcare tech and digital measurement tools, that there's sort of interdisciplinary teams, and not everybody has like advanced scientific training, nor do they need it. But one thing that I definitely take for granted is like I was taught and trained as a scientist to be very aware of such biases and to always be working to try and disprove my hypothesis rather than the other way around, so that I can alleviate any sort of human error that I'm introducing. But often, at least some of the folks that have been on my team of younger data scientists or interns or graduate students often will need these kinds of reminders because they're so excited to see the results that they want to see. It can introduce these kinds of biases. I think this is really helpful information for, particularly in our industry when there's a lot of like software developers and engineers and people that didn't traditionally come from like scientific research or healthcare research, but are really talented data scientists and software engineers are doing healthcare research now in a lot of these like smaller companies. It's really helpful to have this kind of information ingrained in those teams and in those processes as well.
And I'm going to riff off that if I can. I have a very distinct memory of something that feels like 1,000 years ago now. But when I was, working in health services research, there was a big initiative at the particular health system where I was working to try and keep people out of the emergency room. And it was a very, very big, expensive initiative that was launched. And the data was showing like nothing to bear it out. And the number of times the two clinician leads went back to the statisticians to like, well, what about this subgroup? What about that subgroup? Like really passionately, because they were trying to help. And, the intervention was designed with the best of intentions, sort of grasping at trying to sort of see, glimmers of hope that it was actually sort of having an impact there. I remember it really, really well. And that was sort of challenging at the time. A question I have for you all is, it seems to me that these two pieces, right, sort of conflating statistical significance with sort of a finding of actual sort of significant interest or importance clinically, and this idea of sort of harking, I love it, and sort of, getting ourselves to a point where we're actually just seeing signal in what was noise. It seems like these two things are potentially have greater negative impact in the digital era, where there's a lot of data and a lot of noise, and the ability to sort of say, this must be novel, not inconsequential. Like, I wonder how we stop this from sort of derailing forward progress and actually focusing our attention in the wrong areas. Like, how do we sort of communicate all of these things as concepts to everyone, from sort of data scientists themselves all the way up to investors who should be looking at this sort of thing?
Yeah, I can start to answer in the sense that I had, as I progressed in my career, I tried to use language for certain audiences. So like the artificial intelligence piece, I'd written it really thinking about harking at the beginning, but as the piece progressed, it turned to, I should be talking to data scientists like me in my field, hence the title Artificial Intelligence, because it plays on artificial intelligence. And I started changing my language at the end of the article from confirmatory analysis versus exploratory analysis to confirming insights versus discovering insights. So maybe part of the answer there, Jennifer, is, at least that I take, is to use the language of the audience that you're trying to speak to.
Yeah, and I think that gets back to the, like, I'm conscious sometimes when I know there's a data scientist or a statistician, because I will talk about, like, I'll catch myself and not say accuracy. I'm like, it's not correct. And I think that that intentionality around language is actually really important in our field, sort of more generally. Michelle, you teased out something from Eric's articles that I think really resonated with me as well. Do you want to unmute yourself and give voice to that comment? Oh, Smith, you're moonlighting as Michelle. Hey, Smith, do you want to? It was a great point. Do you want to raise it with Eric?
Absolutely, Eric. Thank you so much. This was fantastic, and this was bringing my nerdy statistic class memories from school time. So I'm really enjoying this. One of the things that as I was reading this article, I really appreciated the fact that how we see things in black and white and how we see things in specific orders and standards. One of the responses that you had to this tweet of like, there's no such thing as E in evidence, like there's T in temperature. Wanted to hear your thoughts in terms of sharing on one, why is that? Yes, we see that statistical evidence in terms of how we standardize things and not have that gray area, but also if we are trying to take this back to more, as Jen was mentioning earlier, action-oriented deliverables and action-oriented next steps of aligning it to different stakeholders so that they understand and they start utilizing this language, what message you would like to share with them? Say if it's regulatory, say it's payers, or say it's someone who is actually evaluating that evidence.
Yeah, yeah, thanks for that, Michelle. I'd like to, pardon me, I'm not being rude, I'm just pulling up the actual pieces on my phone so that I don't have to switch screens. I think I had tried to write some terms in to substitute for, you know, significance, so like discernible, inferable, apparent. I personally, I would love to see regulators, excuse me, and other stakeholders start to use those words for the actual finding itself. So this coefficient is discernible or inferable. You know, just a few days ago, I thought, oh, precise might work, but there's a little more nuance to it, and precision has a technical meaning. In statistics, I don't want to confuse it anymore. But I think like you could, my sort of wish list, my incremental wish list would be, you can still use significant or statistically significant, but use it to describe just the p-values, and then say, but the finding is discernible or it's not. The next incremental step would be start to lose the boundary itself, because those are problematic. And there are many wonderful authors in these two American Statistician issues that have written about why that's the case, why putting a boundary on it is not the case. But bringing it back to your point about stakeholders and needing to make decisions, that's also not that feasible with you know, like a regulatory body or a clinical body or a group that has to make a decision. That's, I think, why, you know, 0.05 and 0.01, these came up to begin with, is because we just need a cut point. We know yada yada, there's, you know, there's uncertainty here, but to make a decision to move, we need to have some kind of cut point. The hand wavy wishy-washy answer is that there's an entire field called decision theory that's adjacent to statistics that's, I think, might have better answers for how we think of decision-making rules versus like this statistical cut point, which originally was really just meant as a guideline, not as a definitive, okay, now we're going to push this out to all the patients. You know, also, yeah, I think, you know, it makes me think of what Vladimir had asked earlier. Another change in practice would be, at least for data scientists, with this overabundance of riches, to parse out your data so that you save certain holdout data sets, and maybe you even pre-register those and say, here's the holdout data. And it's trickier to, I don't have a good answer for showing like, oh, here's our evidence that we analyzed, we took our model and we tested it against the holdout data the first time, and you can tell it's the first time versus, you know, multiple times after. I don't have a good answer to that, but Jennifer and I were talking a little bit, and I think there might be some headway there from the engineering side of data science, where they have production pipelines that are highly auditable to see, you know, their timestamp to see like when you made certain decisions. I'm thinking like Jira, Scrum, any of those agile systems. But anyway, yeah.
Very interesting.
Yes.
Really appreciate it.
Thanks. Yeah, and I also I love that from this this Twitter friend, I guess, of mine who wrote this. I'm going to quote that. I told him I'm definitely going to quote you. So be prepared for some hat tips, some acknowledgements.
I thought this was, I mean, I think this is really important. And one of the places my mind goes, and I've worked closely on some of these concepts, Jessica and Kate, I can see you are on the line. One of the things that we bang the drum on pretty hard at DiMe when sort of developing these new measures is to actually do the V3 process first. So obviously we do our sort of verification, our analytical validation, but then really importantly is the clinical validation step, right? Which let's establish the fundamental meanings of this measure. And part of that is understanding what clinically important change is in a given population. Like, to me, I actually think there are fundamental things that we have to continue to press for when we're looking at these digital data, to stop the sort of the conflation you're talking about, Eric, making sure that we actually do that clinical validation first and intentionally and separately before taking the step to then use that tool to answer questions. And what I see a lot of is building the tool while testing the tool. And I think that that could be contributing in the digital space. And Jessica, Kate, I don't know if you're somewhere where you can weigh in on that, you might not be, but that always, you know, that's sort of been emerging through the course of the conversation today.
Yeah, I can chime into that point. That was, I really liked what you said, Eric, about not having great tools for exploratory data analyses. And I think that kind of goes hand in hand. I think many of us who have training in science and statistics know the best way to do something. And that links in with what you're saying, Jen, you know, follow V3, get everything figured out first, then go put it out in the world. But the tech timeline is like, okay, let's do everything at once. And we're collecting data, we're exploring the data, we're testing it, we're kind of trying to do everything simultaneously. And I think, yeah, that really resonated, that we don't have the greatest tools for exploratory analyses. I like what you said about holding out some of the data so that you can kind of do internal replications, even in your own exploratory data sets. But I think it's true for V3 as well. I think a lot of people, the framework is really, really helpful and people see it, but then it's like, okay, but push comes to shove, we have to, you know, push this out there faster than, or it's too expensive to collect all this beautiful validation data before we go out there into the world and start trying things and that sort of thing. So I think there's a little bit of a collision of best practices and reality that can put us in tough spots as researchers. And that's an important point to acknowledge, and still think about how can we still have best practices even if we have some of these real world limitations.
Yeah, and I'll just add to that. I couldn't agree more with sort of when push comes to shove, right? Like, we all know the proper way to do it, but, you know, clinical work on a tech timeline like that's pretty difficult, as said, when push comes to shove. And I think another thing that very much resonated with me, Jen, was your comment about is digital measures, are we more susceptible to these sorts of failures or limitations here? And I think the answer is absolutely yes. Like, we're in a position where we can collect massive amounts of data, right? And like, when we can collect data on hundreds of thousands of people for, you know, long periods of time, it's sometimes more difficult to find non-significance than significance. And so that's really important to consider, right? Like, of course something was significant, but what does it mean? So, I mean, yeah, I think definitely we just collectively as a community need to continuously be evaluating, is this measure, is this tool, is this finding relevant? What does it actually mean? And I think, yeah, we're all excited to move the field forward, and it's much harder to take a step back and really say, you know, is this meaningful than, you know, to just push ahead. So I agree.
I think a positive version of that too is that we're building tools and measuring things that haven't been measured before. So, you know, in some of our work, it's like, okay, well, what does the literature say? And there's many cases where it's like, well, no one's looked at speech patterns in this patient population, so there isn't literature. So we can't a priori define 3 variables and only check those. We have to kind of go exploratory because people aren't measuring this. And so that's exciting, but then it presents challenges statistically because you can't have these really nice pre-registered hypotheses and defined variable sets and so on.
Yeah, I'm loving this back and forth. Thank you for your input, because I have 2 big thoughts that really connect with this. One is like, yes, totally. Like, I think one of the big messages is destigmatize exploratory analyses. Like, literally, this is what science is, you know. And I think to get to kind of more how to do that, I think like one way to would be, as we've been talking about, would be to like parse out, you know, if you have a large data set and try to do replications, like you were saying, Jessica, do replications just within the data set that you have. But maybe even if that's a big lift, yeah, I've had to design studies for some clients where it really is exploratory, and we say that out the gate, it's exploratory. This doesn't invalidate p-values and confidence intervals and statistical significance. All those tools still exist and they're great. They just have a different meaning, right? And you get things like p-hacking and harking because those the meaning is like conflated with the original use of the tool, which is for let's confirm if A versus B is bigger, you know. So I think one of the low lift versions of moving forward would be totally, let's keep using the tools we have, but we state that this is exploratory. So that will trigger us to then want to collect more data. The second big thought I had was, and I would love all your thoughts on this, but I think I learned this lesson through the pre-mortem actually at DiMe, was how do we, maybe using the language of risk to the clients or to your PI or whatever would help more. And it's like the stick versus the carrot, where we say, if we don't report that this is exploratory and you roll this out to your customers or your clients or your patients, you know, you could end up, it's a potential harm in clinical trial terms, because you could end up putting out this program that, you know, usually it takes a long time in science and clinical trials because they're FDA regulated. But if we do it in the wellness space or health space where we can change our app, you know, configuration like that, it might end up harming people because we thought it was true for everybody after we kept fitting and fitting and fitting and fitting. So maybe using the language of risk and harm might actually help. Yeah, would love everybody's thoughts on that.
So I'll jump in real quick if I can. So Eric, I couldn't agree more with destigmatizing exploratory, right? Like I'm the biggest proponent of exploratory studies at this point in, you know, digital medicine, right? We're at a point in this space where discovery is just waiting to happen. And I think that when we don't do these exploratory studies and analyses, we're really sort of limiting ourselves. And in my mind, yeah, it's such an exciting time to be discovering things in this field that let's promote exploratory work. Yeah, I'm in very much agreement with that.
And Kate, really quick on that too, when I said like the metrics still hold, really, I mean, the interpretation only changes very slightly. It just changes to, okay, all these things that we explored and we discovered, right? If we were to run another study or get more data on it, these are about the fuzzy bounds we would expect to see. That's it. You know, it's nothing too wild. So yeah.
And these hypothesis generating studies are so valuable, and they inform the future studies going forward. And that's the way to really develop these measures in a meaningful way on a foundation, you know, that is strong and can withstand the test of time, essentially. So.
Yep, agree.
Hi, this was a terrific discussion. There's a couple of things I really enjoy here, which is sort of the enthusiasm for where we are with the field. I think the possibility of sort of reining this in and getting this right with a few intentional steps, you know, describing specifically what it is we're working on, being intentional with our language, which, you know, is a drum we bang often here at DiMe, and also, you know, I think some education of sort of different stakeholders in the field to be able to discern sort of what they see when they see it. But enormous opportunity here. Eric, this was a tremendous discussion. Really appreciate you joining us today. Two tremendous articles. I think we heard from Danielle sort of the value to a variety of different readers in this information. Eric, thank you for joining us. Everyone, thank you for participating in such a great discussion. I certainly learned a lot and hope that it was valuable for many of you. One last thing before we wrap for today, our next event here at DiMe is actually looking at patient adherence and activation, or patient engagement and activation for better adherence using digital platforms. That's scheduled for Wednesday, September 15th. And as we said at the beginning of this call, both the recording and the slides from today will get posted on the DiMe website, as well as ongoing links to the two blog posts that we discussed today, Eric. They are really tremendous pieces of work. They were very accessible, and this is the kind of thing we need to do to affect change. So thanks so much, Eric. Thanks so much, everyone. Have a wonderful day.
Thank you for having me. Thanks, everyone.
Bye-bye.
Thank you.